So our next talk, we're going to hear about ongoing work, I think, that's hot off the presses on the CMT-SORD, Govorestat, I'm sorry, for CMT-SORD, for the SORD trial. So I'm really looking forward to seeing the latest data on that.
Thank you, Dr. Abrams. Thank you to the organizer, but also to the two previous speakers, because I think I'll try to echo some of the considerations they made in their talk, the biochemistry of identifying a marker that could be predictor of outcome, and perhaps attempt also to address the issue of selecting the appropriate COAs. So CMT-SORD is a form of CMT that is due to a gene mutation on the sorbitol dehydrogenase gene. Patients with SORD deficiency are unable to transform sorbitol into fructose. They accumulate a very, very high level of sorbitol, and the level of sorbitol that we see in their bloodstream are up to 50, sometimes 100 times more than what we have in healthy volunteers. So a very, very significant increase. Phenotypically, they start off presenting problems with their lower limb mobility. Typically, they are in their second decade of life, and they realize that in terms of their performance with sports, they are not doing as well as the year before, and slowly they progress. This is a slide that shows some of the components of the disease phenotype, and perhaps the most interesting component to me is that there is a staggered, slow evolution with accumulation of components of the phenotype. The bar, the size of the bar also showed the heterogeneity at which some of these components of the phenotype may develop over time. So this type of information, which comes from natural history studies, is very critical when you design a study on a new condition, perhaps for a first-in-class, in which you may not know as much as you would like to about the component of the disease that are modifiable when they actually manifest themselves, and hierarchically how relevant they are to patients. So here are some of the components of the disease phenotype. So those are some of the considerations you need to be making when dealing with a disease that has not yet been part of the development program. You need to try to understand the best you can the characteristic of the disease, but also how they change over time, and whether any of them will change in a time frame that you, as a person developing a clinical trial, is able to detect. And obviously also how relevant are potential changes to patients. FDA in large part, EMA in also large part, are emphasizing the relevance in selecting study endpoints to consider the clinical relevance to patients' well-being. This is not only out of being aware of the specificity of what patients face. We have experienced cases where instrumental-driven changes were not associated with an improvement of quality of life. We have unfortunately experienced the fact that what we as physicians would have considered a clinically meaningful change was not always associated with a change in quality of life. So we have all to learn from that. And one of the things you could do when you really need to start to look into a box that is still closed is to meet with patients and start asking them what are they experiencing and how the different components of the disease are relevant. Perhaps also what type of expectation they have. Is there expectation that there is no progression? And is no progression a sufficiently acceptable expectation? Is the expectation different? Is the expectation that there is reversal of symptoms? Is the expectation that there is a extension of life expectation? So all of those things need to be considered to make sure that your clinical trial may be clinically meaningful. So going back to the biology, in these patients, because they're not able to process sorbitol, they and able to make fructose out of that, there is an excess of sorbitol. Now, the pathway that is affected is a very secondary, probably an evolutionary remnant shunting of glucose. In humans, we don't use that much. We don't use that for energy production. It's actually a pathway of glucose routing that doesn't yield any ATP. The molecule we're developing is a molecule that blocks the making of sorbitol, so target an enzyme that is one step earlier than the one that is defective in this patient, target an enzyme that is called aldose reductase. When designing the clinical study, we had to deal with the complexity that, although from a biochemical point of view, we felt that there was a good understanding of the biology of the disease, also supported by two independent animal models, a Drosophila model and a rat model that showed that the gene mutation was responsible for increased level of sorbitol and for development of the phenotype. We were also in a dialogue with the agency to discuss what is relevant. So what you see in this schematic is a study that is a folding of two components, one that is pure biomarker and one that is clinical outcome, because we felt that sorbitol could have been the reasonably likely predictor of clinical outcome, but we also needed to add additional validation component to that outcome. And we felt that perhaps if we were going to see directional changes in some of the clinical outcome, that could have also support the validation of sorbitol as a clinical as a biomarker. So the study was evaluated at 3 months from a biochemical point of view, at 12 months to see whether there were directional changes of some of these clinical outcome, and planned to be completed at month 24. The baseline is also very interesting. We conducted analysis of the baseline in light of trying to further validate the case for sorbitol. So what we have demonstrated is the baseline severity of the disease as assessed by the CMT form, correlated with the level of sorbitol for what concerned the lower limb domain. So the components of the CMT form or lower limb domain had a correlation with the level of sorbitol. Patients with the highest level of sorbitol were more severely impaired, and obviously patients with a lower level had a more mild disease. On month 3, there was an evaluation of the level of sorbitol. As predicted, patients exposed to active treatment had a large reduction in their sorbitol level. The drug was generally safe and well tolerated, and there were also changes in NFL. They were very promising. At month 12, we conducted an interim analysis where we analyzed the relationship between a composite of the clinical endpoint of 3 components: 10-meter walk run, dorsiflexion, and 6 minutes walk. And we assess whether changes in the combined endpoint related to the level of sorbitol. What you see on the left of this slide is that favorable changes in this composite endpoint correlated with the level of sorbitol. So patients with lower level of sorbitol had a more meaningful change for a composite of these 3 endpoints. However, the study primary endpoint at month 24 is not this composite, but rather a single component of CMT foam, which is the 10-meter walk or run. And what you see on the right part of this slide is the correlation between changes in 10-meter walk or run and sorbitol level. So once again, lower sorbitol level is associated with a more meaningful change in 10-meter, more favorable change in the 10-meter walk run. The CMT high, which is a patient-reported outcome, you heard that in the previous talk, showed very meaningful change, and they were very systematically affecting all of the domains that were considered in the CMT form. What you see first in this slide is the total score, which was statistically significant. And what you see below are the different components of the CMT-I, the lower limb domain and the upper limb. Then you see fatigue and pain. It seems that generally speaking, there is always a trend in favor of the treatment versus placebo. What you have in this slide is this overall safety. The drug was generally safe and well tolerated. The events were generally mild or moderate. The study is obviously overseen by a drug monitoring committee that reviewed the safety in an unblinded fashion periodically. So in general, I think what we have seen that Govorestat produces significant lowering of sorbitol. The level of sorbitol over 12 months are a predictor of changes in the CMT-I, the patient-reported outcome. We also see a relationship at month 12 between the level of sorbitol and a composite of the clinical outcome measure. Thank you for your attention.
Thank you. We have time for maybe 2 or 3 questions, actually. So I will start. I have a question. First of all, I want to apologize for not introducing you, Dr. Perfetti. I think I just gave the topic of the talk, so thank you very much for the talk, and I apologize. My question is, did you have an a priori assumption about what would be a biologically significant reduction in sorbitol in these patients? And if so, do you think you've met that a priori assumption?
That's a critical question, right? So these patients have levels that are astronomically higher than healthy volunteers. Do we need to take them to the level of healthy volunteers? The animal model suggests that level of reduction greater than 30% in the knockout animal model are sufficient to rescue the phenotype. Interestingly, we were able to assess the sorbitol level in some of the heterozygous relatives of the patients that are in the study, and very differently from what I would have assumed, their level was significantly higher, even tenfold higher than healthy volunteers, but they had no phenotype. So what that suggests to me that we humans have a biological tolerance to accumulation of sorbitol that at some point goes over a threshold and we develop a phenotype. That's my assumption so far.
Are there any other questions at this time? We'll have more opportunity.
What natural history data was available for SORD, and if there wasn't any, did you have to put that together in order for this to be successful, or at least demonstrated successfully?
Because CMT-SORD is a recently identified subgroup of CMT, the natural history data are still very limited. So that's why in the discussion with the FDA, we end up having this very large bucket of endpoints that are biochemical, clinical outcome, patient-reported, for two reasons. One is because you like to know the most you can, but also because, very honestly, we didn't know what to look for. So we wanted to have a permissive approach, obviously pre-specifying the analysis. But to answer more directly your question, unfortunately, the natural history data on CMT-SORD are still a bit fragmented.